Welcome students. My name is Irfan Ahmed and today we are going to study about the topic that is transport across cell membrane. So in the last topic we had studied about the transport. Now we will continue from the same. So passive transport we studied in the last topic. So now we will study about the active transport. Now what active transport is? So active transport, it is another type of movement. It's also based upon the concentration gradient. So passive transport, that was also based upon the concentration gradient, and this type of transport, which is active transport, this is also based upon the concentration gradient. Okay, so what is the difference between active and passive transport? So main difference is that in the active transport, materials move from region of lower concentration to higher one, where in that of passive transport it occurs from higher to lower. Okay, so that is just the opposite. Active transport is just the opposite of the passive transport. So in passive transport no energy is needed, while in active transport we need energy to carry out the active transport of molecules. Another difference which is in the active and passive transport is that energy is required in the active transport, while as no energy is required in the passive transport. Okay, so the energy which is used in the active transport, that is used to push the solutes against the concentration gradient, because we have to go against the concentration gradient. In passive transport, the transport is occurring in the concentration gradient. Here we have to go against the concentration gradient. We have to move the molecules from low concentration to that of high concentration, so we will need energy to push up that. Okay, so there are three types of active transport. We will discuss that. Membrane pumps, first one. Second one is endocytosis, and third one is exocytosis. So membrane pumps, these are carrier proteins, okay, which help in the movement of material from the lower concentration to higher concentration. You have to remember this thing in active transport, that we have to go from lower concentration to higher concentration, where in case of passive transport we were having from high concentration to, in case of passive transport we have to go from high concentration to low concentration, but in case of active transport we have to go from low concentration to high concentration, means we have to move against the concentration gradient, and we will need energy for the same. So the carrier proteins which bind to specific molecules, so high concentration to low concentration, then there will be conformational change, then it will require no energy, and it is selectively permeable. Okay, these are the things which are to be remembered here. Carrier proteins bind specific molecules, okay, and then they carry out the process from high to low concentration. Conformational change will be there, it will require no energy, it will be selectively permeable. Okay, so the example of membrane protein, I will carry out the example of membrane protein, is the infamous and important example of membrane proteins, that is sodium potassium pump. Okay, so sodium potassium pump, what happens in this sodium potassium pump? So outside the cell there is high concentration of sodium. If we take an example of the cell, so outside of the cell there is high concentration of sodium, and while inside the cell there is high concentration of potassium. You have a cell, outside is high concentration of sodium and inside is high concentration of potassium ions, and the function of that pump, it's also always to push sodium outside the cell and more potassium inside the cell, because inside the cell there is more potassium, so more potassium will be pushed into the cell. Outside the cell there is more sodium, so more sodium will be pulled out of the cell. Okay, so the process of doing this start by having 3 Na positive ions binding to the pump. So first of all, what happens, 3 sodium positive ions, these bind to the pump, then ATP molecule is used to change the conformation of the pump, and then one phosphate ion is also attached. Then what happens by this process, sodium positive ions is released to outside of the cell, okay, and 2 potassium ions then bind to the pump, causing the phosphate ion to break, allowing easier release of potassium inside the cell. So when the sodium moves out of the cell, then what happens, potassium at the same time comes into the cell. This is called as antiport. Symport means if sodium is going out, potassium is going out, then that is symport. But what happens there, sodium is moving out of the cell and in case there potassium is coming into the cell, so this is antipode type of transport. Okay, so this is an example of sodium potassium pump which is carried, which occurs in our body. Okay, so outside sodium concentration is more, inside potassium concentration is more. Now sodium moves out of the cell and potassium comes inside of the cell, and for this what ATP is used. ATP and adenosine triphosphate is used to carry out the change in the conformation of the pump. Only then the phosphate ion can attach to the pump. You can see here, this is an example. For example, this is the, I will now give an example. For example, this is a high concentration of sodium, this is the potassium. By this step, this is extracellular fluid, this is cytoplasm. So potassium will move inside the cell and sodium will move outside of the cell. So now there are two main types of active transport. These are, one is primary active transport and secondary active transport. So two types are there, primary active transport and secondary active transport. And the difference between these two again is on the basis of the energy source used to drive the transport. So the difference between the primary active transport and secondary active transport is only because there is a difference in the energy source which is used to drive the transport. Okay, so in case of primary active transport, the driving force, the energy source, that is adenosine triphosphate ATP, which is also called as the energy currency of the cell. So ATP is used to drive the movement of the solutes. So the solutes can move only because of the ATP. While another case is secondary active transport, which uses other sources of energy other than ATP. The sources of energy which are, those are used in the secondary active transport. So primary active transport will entirely depend upon the ATP, while a secondary active transport will depend upon the secondary modes of energy other than the ATP. We have given the example of a sodium potassium pump before. Okay, so in the process, after the transfer of lot of sodium ions outside the cell, when the lot of sodium ions move outside the cell, then what happens, the outside of the cell membrane, that becomes more positive because sodium is a positive ion, sodium is a cation. Okay, so now here comes the principle of electrostatic repulsion. Electrostatic principle of electrostatic repulsion, what it says that opposite charges repel each other, then an electrochemical gradient is formed in this process. This is the principle of electrostatic repulsion, which says that opposite charges repel each other and electrochemical gradient is formed in the process. Okay, so this electrochemical gradient which is formed in this process, it is a source of potential energy. Potential energy also because of the position. So it will drive the movement of any positive ions outside the cells, okay, to enter the cell once more to reduce the positive charges outside, because outside the positive charge has become more. Okay, now this electrochemical gradient, it is a source for potential energy that will drive the sodium ions outside the cells to enter the cell because it has to maintain the charge. Okay, so the movement occurs through a symphotor. What this symphotor does, it uses potential energy due to electrochemical gradient which was formed. Okay, so in terms of sodium ions, the movement is from higher to lower concentration. Now, however, this process is also considered active transport because aside from the sodium positive ions entering the cell, glucose also moves from the outside to inside the cell. Now glucose is also moving, so glucose concentration outside is lower as compared to inside of the cell, and this process will use the energy, and this is also called as active transport. So this is also the example of the active transport. Now you can see here what happens. Three sodium positive ions move out here. Three sodium ions move out. Then what happens? Two potassium move in. You can see here the potassium moves in. Then what happens? Then ATP is hydrolyzed. So ATP process in whole process the ATP is used. So main thing we have to remember here is that the use of ATP. So ATP is hydrolyzed. ATP is used. So here ATP is used, ATP binds with the phosphate, then 3 sodium positive ions move out, then 2 potassium ions move in, because when sodium moves out, potassium moves in. So here potassium moves in, here is phosphorus, okay, then this happens, and then ATP is hydrolyzed. This process gradient. This process is repeated again and again. So one more type of transport is bulk transport. Now what bulk transport means? When a lot of materials are transported at a once, at the same time when a lot of materials are transferred, then that transport is called as bulk transport. Okay. For example, vesicle movement, like for example there is endocytosis, there is exocytosis. These two transports are called as bulk transports. Okay. So there are two types of vesicle movements which occur in the cells. For example, endocytosis is there, exocytosis is there. Endocytosis is, it brings items inside the cell, where exocytosis pushes items outside of the cell. You can see here one example is phagocytosis, cell eating. So here the cell membrane changes its shape and into the vesicle, it is called as cell eating. And this is pinocytosis, this is called as cell drinking. Here you can see the solute molecules goes inside the cell, a vesicle is formed. So this is a process. This transport is a bulk transport. Okay, so endocytosis is an active transport. It uses energy to take biomolecules or larger particles by the formation of the vesicles. It also involves ingesting external fluid which contains macromolecules, larger particles, and this works by having the cell membrane that is a pinch around the material, formed a vesicle. That vesicle will carry the substance in any part of the cell. So two types of endocytosis are there, phagocytosis and pinocytosis. And phagocytosis again means when large molecule materials are ingested at once. And while pinocytosis means finer, smaller molecules are ingested. So commonly phagocytosis is termed as cell eating and pinocytosis is termed as cell drinking. In phagocytosis solid particles are ingested, in pinocytosis liquid particles are ingested. So this is about the phagocytosis, pinocytosis and pinocytosis, which are the types of endocytosis. Now second one is exocytosis. It involves vesicle containing the material fusing with the cell membrane. Means vesicle material these fuse with the cell membrane. Because of this material is released to the outside of the cell. So this is used by secretory cells, this type of transport. This is used by the secretory cells, this is used by the neurotransmitters. And because these two processes require energy, they are the forms of active transport. So these are also the parts of the active transport: endocytosis, exocytosis. So endocytosis includes phagocytosis and pinocytosis, while exocytosis it is the fusion of the membrane with the material, then transporting the material outside of the cell. Okay. So this was all for the today's lecture. In next topic we will study. In next lecture we will study about the next topic. Thank you.